check it. Check it. Oh, I think I got the fever. I'm heated, pumping up the meter. Roll me a ringleader, mad party bleeder. Make this with a storm, lead after lead after leader. Hey, can you dig it? I come alive, fella, give me what you got. Back up, pump, stick up, pump your fist up. You check the shadow, no buckshots, a bus slip up. Morning like a kid, gonna make it feel alright. Pump it up all night, body rock, show me what you got. Let Rob break the moves, slip up. Morning like a kid, gonna make it feel alright. Pump it up all night, body rock, show me what you got. Let Rob break the moves, Do you wanna get check it out, check it out, check it, 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 check it out. Sometimes I feel like I don't have a partner. Sometimes I feel like my only friend is the city I live in, the city of angels. Lonely as I am, together we cry. I drive on the streets 'cause she's my companion. I walk through the hills 'cause she knows who I am. She sees my good deeds and she kisses me when and I never worry. Now that is a lie. I don't ever wanna feel like I did that day. Take me to the place I love. Take me all the way. I don't ever wanna feel like I did that day. Take me to the place I love. Take me all the way. Yeah, yeah, yeah. It's hard to believe that there's nobody out there. It's hard to believe that I'm all alone. At least I have her love. The city she loves me. Lonely as I am, together we cry. I don't ever wanna feel like I did that day. Take me to the place I love. Take me all the way. I don't ever wanna feel like I did that day. Take me to the place I love. Take me all the way. Yeah, yeah. Oh, no, no, no. Yeah, yeah. Love me, I say, yeah, yeah. One time. Is where I drew some blood.
Hmm. Hmm. All right, let's see who's here. Huh. I don't know. Who's here? Can't see the chat or the participants. All right, I'll give it a couple more minutes for people to get here. Bryce. Okie dokie. It's time to get started. Alrighty. Good morning, everybody. Good morning. Good morning. Uh, this is the last day before um, spring break. There's no school tomorrow. So I already gave you guys the Unit 18 test. Luckily, we were able to do that before break. And now I wanted to kind of approach just a novel topic. It's not by any means something we're going to explore in depth, but I think it's appropriate considering the circumstances. Um, so yeah, let's just dive right into it. Let me change my stream really quickly. There we go. Okay, so we're going to be talking today about viruses, and I'm pretty sure you, I'm pretty sure you guys already saw the worksheet, since I gave it a period 3 yesterday. The worksheet should be fine now. I'll pull up the Google Classroom page to show you. So I'm not sure what happened the first time I posted it, because I put it up and I allowed you guys to edit it because I know some of you guys go into Google Drive and you guys like to just put the answers right there. But I'm not sure if you were actually editing the attached file. Maybe someone erased the whole thing. So I put up. I gotta start putting up PDF versions because they'll show up the way they're supposed to. Yeah. Then when I uploaded a document version, like the text wasn't here in these boxes, the arrows weren't there. It just looked really nasty. So I'm going to be uploading PDFs, which might make it difficult for those of you who do it on Google Drive. But so yeah, it's here. It's fine. It's done. No problem. So you can just do it from here. But let's continue. Let's continue and talk about viruses today. A virus. Um. Uh. And we will briefly approach the COVID-19 topic. There's an interesting video I think that we should all watch as sort of a way to recap the way that viruses work. Um, oh my goodness, not many people are here right now. It was erased. It doesn't. Please open in Google Docs. It's a PDF file now, Bryce, so you should be able to download it as a PDF. It may not allow you to open and edit it in Google Drive, but it's definitely not the messed up version if you go onto the classwork tab right now. Good morning, Connor. Good to see you, dude. Thanks for the heads up last night. Apparently last night I left my stream on like way too long, and I was just chatting with a friend, and then playing some games. I had no idea it was still on. So appreciate that, Connor. Um, hey Garrett, how you doing? Hey Adam, Adam's here. Okay, people are starting to come in. I guess I'll give it a couple more minutes. Um, and then we can begin. I did not. I'm not going to be posting third period meetings, just because I don't want to post the same thing twice. So, although I recorded the third period's meeting, I didn't record some of the stuff I'm going to show you guys today. So, I will definitely be showing you some cool things. Respond to your email, Garrett. Okay, Paris emailed me. Yeah, okay. Alright, let's see who's in the chat now. Let's actually just do an attendance check on the Google Classroom page then, yeah? That seems like the best thing to do. So let's go over here. Attendance check. I want you guys to put down your favorite Christmas movie. Your favorite Christmas movie. Actually, maybe not Christmas, but holiday movie. How about that? Holiday themed movie, regardless of what holiday it is. I'm gonna put How the Grinch Stole Christmas because that is indeed my favorite holiday movie. Not the Grinch, not the animated one, the original one with Jim Carrey. That one takes the cake. I do type fast. I'm an awesome typer. Garrett, when did you send me an email? I get emails all the time. Which email? You want to just type it in the chat? What's the deal? About the science fair? Okay, let me check. Garrett. Can you resend it? I get like 500 emails a day. I'm trying to find it. All right, people are commenting. I don't really watch movies. Okay, how about then, Connor? Go ahead and put your favorite superhero. How about that? You could do your favorite superhero. Garrett. Can't find the email that you sent me. Is it possible to resend? Yeah, resend me that email, please, Garrett. Okay, I'm just gonna get started now, regardless of who's here. And yeah, let's get down to business. So, Garrett, you could just send it to me as we go along, okay? Let me start recording right now. No, that's not my recording tool. Start recording. Okay, so we are talking about viruses today. Uh, Viruses are incredibly interesting in both their, I guess you could say, ecological role. They do have one. Despite not being living, which is the most craziest thing of all, is that we do not consider viruses to be living things. If I were to show you guys a list of characteristics that viruses share with living things, it would be well, let's just do it right now, okay? Um, the basic characteristics of living things are as follows. Living things, for one, they reproduce. Sorry. Let's start from the very basic. They're made of cells. All living things are made of cells. Okay. All living things move and use energy. All living things have DNA. Good morning, Sammy. All living things reproduce. Oh, sorry, DNA or some other genetic material, right, for passing on hereditary information. Uh, all living things respond to stimuli. When I say respond to stimuli, meaning if there was a change in the environment, they would respond to it. Um, and that is both long-term and short-term. So if someone chucked a ball right at my head, I'd respond to it if I noticed it. If it stimulated my photoreceptors in my eyes, I would respond to that stimulus by getting out of the way. And then there's long-term responses to stimuli, which is basically just evolution, law change over time. So you can respond to something like the climate changing. You can respond to changes in the average temperature of an area. Uh, Long term, of course, but nonetheless, it's still a response. Just one that takes a really long time. Let's see. What's the last one? Made a cells when used to reproduce, respond to stimuli. Oh my God, I forgot that last one. Stimuli. I'm blanking on the last characteristic of living things. Well, they have some sort of metabolism. I guess that describes using energy as well. So we'll just put that here. Uh, they use energy. I know movement requires some form of energy as well, but the movement can also be used to describe sort of the movement of materials in and outside their cells. That's another way to describe it. And even better than the list I've made up here, there's a list further in this section I can show you guys here. where it compares a bacteria to a virus. And it's right here. Let me get this out of the way. Uh-oh, out of the way. There we go. So here are the comparisons between a virus and a cell. Now, obviously, a virus is not a cell, so it cannot be made of cells. This is the one characteristic, well, one of the few characteristics that viruses do not meet when it comes to describing a living thing. Okay. So they don't have any growth or oh, sorry, yeah, that's the right one. That's the right one. They move and use energy. Uh, they they grow and develop. So that those are the six characters. That's the last one I was missing. So viruses don't really grow or develop. They are assembled, constructed, kind of like a machine would be. They do respond to stimulus, right? They do reproduce, albeit they reproduce only inside another cell. Well, not another cell, a cell. They have DNA. Most of viruses have RNA actually, but DNA is also found in living things. So that's one thing they share in common. They don't really move or use energy. What they do is they hijack other cells for their own and use their energy and resources, and they don't really move on their own. They are transmitted through different vectors and vehicles, animals or mechanical forms of transmission like moisture in the air, wind and water movements and stuff like that. That's how viruses move. Not on their own though. So yeah, they're not by our standard definition of living thing, living. All right, that's one thing that really sets them apart. And it wasn't until recently that they were actually discovered. You know, imagine for most of human history when people got sick, you know, you had no idea, no way to tell uh, what was causing them to be sick. But then you develop microscope technology, and people still get sick, and you can't see what's wrong with them. Well, microscopes are good enough to spot out small bacteria, and bacteria are already pretty a lot smaller than normal eukaryotic cells, and viruses are even smaller. So it wasn't until 1890, well, in the 20th century, we actually discovered viruses. Although signs of them were definitely picked up on before that. So it was in 1892. Let me change the color here. It was in 1892 that this Russian guy, Dmitry Ivanovsky, was studying a disease called tobacco mosaic disease. And this is what the disease actually looks like. It causes these sort of different weird sort of blemishes and weird patterns in the pigmentation. Basically, the cells in these tobacco leaves are dying, and those white areas are the places where the cells are dead and you have lost, are no longer producing pigment essentially. Uh, on the leaves back here, you can see that the blemishes are actually turning brown and dying off. So these tobacco leaves are infected by tobacco mosaic viruses. Okay. Now, here is okay, so let me get this straight. So he was studying tobacco mosaic disease, but he didn't know what was causing it. But he took liquid from the infected plants. He probably mashed it into a pulp and extracted all the liquid material he could, and he was able to transmit that disease by exposing unaffected plants by with that infected liquid from the diseased plant. But yet he could not find the pathogen under a microscope. It was still too small. And there was a Dutch scientist who thought that there may be even like really, really tiny particles, like smaller than bacteria, that were causing some diseases. And that guy was right here, Martinus Bierink. I think that's how you say his name. But he was the one who actually came up with the term viruses. Before him, we didn't really have a word to describe it. You can imagine there is something that you cannot see causing these awful, awful things to happen. And you think it's like a curse or some sort of I guess religious verdict or something on a person's life. But clearer heads prevail in this case. So yeah, uh, the general scientific community accepted the term viruses, and we still use it to this day. And it's actually Latin for poison, although it's probably closer to a poison than it is to a living thing. Actually, no, it's not. Alrighty, so in 1935, Wendell Stanley, this American biochemist, he was able to make crystals of the tobacco mosaic virus. Let me look it up and see what they look like. Tobacco mosaic virus crystals. They probably don't look like anything special. And I cannot find it. Yeah, I don't know what the crystals look like, but he was able to crystallize them. And living things do not crystallize. It's why it was one reason. The reason why is because we're filled with water, and when water crystallizes, it expands, and it'll rupture membranes and cells and things like that. So we can't really freeze things to preserve them. Our stuff doesn't crystallize, but viruses do crystallize. At least some do. Um, and when it was taken out of crystallization, when they probably uh, put it into solution again and took it out of its solid state, um, put it into an aqueous state actually, it still functioned. So it didn't kill, quote unquote, kill the virus, which is interesting. Living things don't do that. Uh, so what is a virus? It's basically just a packet of protein, a packet of protein, sorry, it's a packet, it's a protein packet of nucleic acids, and sometimes it has some lipids in there. Sometimes the virus produces like chemical messages with the lipids, or with steroids or something like that. And they can only reproduce by infecting living cells. It's the only way. They're unable to reproduce independently. So they need to get inside living cells. They need to infect living cells in order to make more of themselves, which is really the virus's only goal. Okay, so let's go ahead and look at these diagrams right here, of three different viruses. We have a T4 bacteriophage. Now this is familiar because we talked about this in our DNA section. This is a virus that attacks bacteria, and well, I mean, we've already examined it. It's got its sheath, it's got its surface proteins. This entire outer surface and these legs are probably constructed of very specific complex proteins. And inside, it's got its DNA core. Um, Thanks, Bryce. The T virus. I wonder what the T virus would look like. Interesting. And then we have the influenza virus, which looks familiar. And I'll show you guys a picture of something that looks familiar in it shortly. And then here is the actual tobacco mosaic virus, and it's got this really cylindrical shape, and RNA is ejected out of the top end of it. But there are some similarities that all viruses share. Number one, all viruses have this capsid, this protein coat. Okay. Now I know it's not labeled here in the bacteriophage, but I guarantee you this whole outer surface, the actual structure of the virus is composed of an outer layer of protein. So they have this protein coat, and then they have these structures on their surface. We call them surface proteins. If the surface proteins are the same as the actual capsid itself, then we just call them capsid proteins. Okay. You notice how the capsid on the influenza virus is a distinctly different color than the surface proteins that are purple and look like plungers. So sometimes there are separate proteins that make up the surface protein. Sometimes the surface proteins are the actual protein coat or the capsid. It really depends on the viruses. Uh, and then obviously the shapes of all these three are very different. We have a lot of different types of viruses, and we create virus families to describe those that are similar. Now, I'm going to go ahead and show you an image of coronavirus. And you guys tell me which of those three viruses I showed you it looked like. To give you guys some time, here's the three the three different viruses that the book shows us: the bacteriophage, the influenza virus, and the tobacco mosaic virus. So, which one does coronavirus look most closely like? The influenza virus, exactly, Bryce. Okay, and why? Because influenza is a coronavirus. I've explained this to you guys before, and I'll explain it again. Coronaviruses are just endemic in endothermic creatures, so warm-blooded creatures. The cold virus, influenza virus, SARS. What else? I can't think of another one. But those three are all they're all coronaviruses. They belong to the coronavirus family, and they're called coronaviruses because they look like coronas, like little crowns. They have these proteins that kind of jut outward, making it look kind of like a crown if you were to examine the silhouette. Yeah, there's multiple coronaviruses. There's a family of viruses that we call the coronavirus family. And many different viruses are in that group, especially ones that affect humans regularly, like the influenza or the common cold. Well, actually, I'm not sure if the common cold is a coronavirus. I'll have to double check that. But here is an actual really good image of coronavirus right here. That was generated using cryo-electron microscopy to generate this shape and then color it. So we'll watch this video here in a bit. But yeah, here is the actual microscopic image with the dyed viruses there. And I'm guessing this right here, this little kind of saggy bag that they're in, is a dead cell that they have just lysed. But let's get back to our lesson. Okay, so a couple things to remember that I want to point out. All viruses will have a capsid or a protein coat. They'll have DNA or RNA inside as like their genetic core, and that's where they'll pass on or they'll use that RNA or DNA to hijack the cell and take it over and start making more viruses. There are surface proteins, and they come in a variety of different shapes and sizes. Like you have these legs right here, but these legs probably only serve to anchor this bacteriophage onto a cell membrane. Uh, these surface proteins are probably used to interact with the protein channels and pumps on a cell membrane. And these capsid proteins, well, I'm not sure. Maybe they're designed to bypass the cell wall of a tobacco plant cell. Okay. Sammy, that's super inappropriate, man. Okay. So let's get on to, well, there's a metaphor that your book gives, which sort of describes how the virus infects a cell. It doesn't. I mean, there's different ways that viruses attack cells, but the book gives this example. The book compares a virus infection to an outlaw in the Wild West that is entering town and taking over the town. So they'll lock up the town sheriff or the town deputy or whatever, and then they'll come in and they'll demand that all the townspeople give the outlaw their equipment and all their resources, and then the outlaw will use that equipment and those resources to create a gang of more outlaws, and then those outlaws will go out to other towns and do the same thing. Eliminate the authorities, to take all the stuff away from the people, and create new gangs. That's similar to how a virus works. Although the metaphor that the book gives is pretty funny. It looks like a Danny DeVito cowboy that's taken over this town. I don't think Clint Eastwood would do this. Sorry, Adam. Okay, let's talk about the two main types of viral infections. We have the lytic infection, or lytic. I think lytic is probably easier to say. Lytic infection and lysogenic infection. Um, and pay attention to the ly beginning to these types of infections. That's important. Okay, so a lytic infection. Uh, a lytic infection a lytic infection involves the virus entering a cell. So in this type of infection, the virus is actually getting inside the cell, um, or sometimes it'll actually just attach to the cell membrane and inject its DNA or RNA into the cell. Now what happens then is the viral DNA is then translated into viral protein. So uh, the genetic material is sent to the ribosomes, the protein synthesis sites of the cell, and the ribosomes will read the viral DNA, make viral proteins, and those viral proteins will chop up the host cell's DNA. And once that DNA is chopped up, the cell is pretty much out of control. I mean, there's nothing, directing it. And now the virus has free range to take over the rest of the cell's organelles and resources and begin making new virus particles. And once enough virus particles are made, the cell ruptures, it bursts open, it lyses the cell. To lyse something literally means to rupture it, break it apart. So that's why we call it a lytic or a lytic infection. And the same thing happens. The same thing happens in the end of a lysogenic infection. The cell is lysed; it is ruptured, and the viruses are able to escape. So let's take a look at the diagram here. There's a diagram right here which kind of walks through both lysogenic and lytic infection types, and it describes where they diverge and where they're similar. Okay, so the first type of infection, the lytic infection, that's what we're looking at right now. A virus, first off, a virus has to attack a cell. It has to come into contact with the target cell that it's trying to take over. So here we have a bacteria cell, and a virus has anchored onto its membrane, and it has injected its viral proteins. Okay, sorry, not viral. It has injected its viral DNA. That's that little squiggly worm thing right there. And once it does that, the viral genes are then transcribed by the host cell, meaning that the virus DNA is actually taken, accepted by the cell, and then it is used to construct new proteins and nucleic acids. Okay. So after the cell has been tricked into making all these new viral proteins and nucleic acids, uh, sometimes what happens is the proteins will literally chop up the cell's DNA. Sometimes it doesn't happen, but sometimes it does. Uh, and then after it's done chopping up all the host cell's DNA, those proteins and nucleic acids assemble into new viruses. Okay? And so many viruses are created that the viruses actually rupture the cell. There's no space, so the cell kind of explodes or implodes, and the viruses escape and attack other cells. But this action of rupturing something and splitting it open from the inside is called lysing. So that's what we're at right here. Okay. So that's just the steps of the lytic cycle. Very simple steps, not too much happening, and it's very fast. The virus doesn't waste any time making those new proteins and nucleic acids. It's trying to construct them as quickly as possible, which is different from the second type of infection that we'll talk about, which is the lysogenic infection. The lysogenic infection, the virus doesn't immediately start to take over. Instead, the virus will inject its nucleic acid. Most of the time, it's DNA. It'll inject and insert its DNA into the host cell's DNA. So whenever that host cell copies its DNA, it's also copying the inserted viral DNA, and the cell isn't damaged at all. But what happens is, let's say an infected cell replicates its DNA in advance of cell division. Well, it also copies the viral DNA, and then it splits into two daughter cells. And then at that point, the viral DNA might activate and begin causing the cell to make proteins and viral nucleic acids. So it's sort of like the long con. The virus attacks the cell, injects its genetic material, and then nothing happens for a while. Sometimes it might take dozens of cell divisions before the viral DNA becomes active. But this is really dangerous, right? Because this can result in you getting sick out of nowhere. You have a virus that has attacked multiple cells and caused them to divide with the infected viral DNA. and then all of a sudden, on the flip of a switch, they all start producing viruses, and all of a sudden you just get sick out of nowhere. So lysogenic infections are the ones that kind of quote unquote come out of nowhere. But yeah, so let's take a look at the diagram. So the only real difference, they all both lytic infections and lysogenic infections end the same way, with the cell being ruptured due to all the created viruses filling up the inside of its cytoplasm. But there's a couple extra steps to the lytic infection that we'll talk about. So we're back here again. The virus has attached onto its host cell, and it injects its DNA. Except the DNA is not immediately transcribed into proteins and nucleic acids. It actually inserts itself into the host cell's DNA. So here is the section of the viral DNA, which we call the prophage, and it fits nicely into the host cell's DNA. And the host cell doesn't recognize this, can't tell the difference. And then it undergoes its own replication. So now the viral genes are copied as well as the host cell's own genes. But still nothing happens. Nothing happens at all. Okay, and this might happen, like I said, dozens of times. You might have many cell divisions, and each of those new cells that are created have that viral DNA in their own genome, which is ready to be activated at any time. So what happens when it activates? Well, when it activates, the prophage actually comes out of the cell's genome. So it leaves the chromosome that it was in, and it enters the lytic cycle. So then it gets transcribed, and it starts to make all these proteins, all these little proteins and nucleic acids in here. And then the same thing happens. Those proteins and nucleic acids are assembled into viruses. Once enough viruses are assembled, the host cell is ruptured and the viruses escape. So that's the difference. They both kind of follow the same end cycle. It's just lysogenic infections take a little more time. And they're sneakier. So it can be tricky to contend with. So if you ever get sick out of nowhere, like I mean literally, just the past few days, maybe you ate something you shouldn't have, and then a few days later you got sick. Well, it could have been a lysogenic infection. The virus could have waited for a few cell divisions before it activated that prophage and started making those viruses. Whereas lytic infections, I remember every time I got food poisoning or something, or some kind of really fast-acting illness, and I could literally trace it back to what caused it, if it was like within the day or within the last couple days, and I know it was probably a lytic infection. But if I get sick out of nowhere, then who knows? Who knows when I contracted that virus? The virus could have been in me for, weeks before it activated, which, it sucks. Alrighty. So let's take a look at RNA viruses. So these viruses, we're describing viruses that inject DNA into their host cells, but there are also viruses that don't have DNA cores; they have RNA cores. And if you don't remember RNA, and what it's used for, basically it's sort of like a really manipulable form of genetic information that can be transmitted and translated from DNA. Okay, so what happens? What are RNA viruses? Well, most viruses are actually RNA. About 70% of them, right here in the book, of viruses are actually RNA viruses and not DNA viruses. In humans, it's RNA viruses that are responsible for the majority of our illnesses, from the common cold to autoimmune deficiency syndrome. So, if you get a virus, typically it's an RNA virus. Okay. Now there's two types of RNA viruses, and we'll talk about both of them. And here is how one particular RNA virus, the common cold virus, operates. So similar to lytic and lysogenic infections, the virus has to get inside the cell or inject its RNA into the cell. So that's what it does. Uh, the difference is, in this particular, in the lytic cycle and the lysogenic cycle, that particular viral DNA, this viral genetic material, there's only one copy of it, and that one copy is transcribed. The same in the case of the lysogenic infection. There's only sort of one set of that viral DNA that is then transcribed. However, in an RNA virus, we have the virus actually makes a lot of copies of its genetic material. So it doesn't release just one single strand of RNA. It releases multiple strands. It starts copying and creating a lot of different strands of its RNA. And well, what happens then? Um, well, since there's a lot of copies of RNA of just floating around, what happens is that RNA is transcribed by the cell's ribosomes, and then new viral parts are constructed. And after they're constructed, the same thing happens. They crowd the inside of the cell and cause it to rupture from the inside, uh, and that's pretty much it. So main difference is the main difference is this particular virus, RNA viruses, make lots of copies of their RNA, whereas a DNA transmitting virus will simply transmit its DNA once, and it'll let the host cell take over from there in translating it and then transcribing it. Whereas the virus just cuts that middleman out, says, hey, you know what, I'm just going to make all these copies of RNA, and they're all going to be transcribed right now. Not, we're going on my time, not your time. Is basically what the virus is saying. Because in the normal infections, the virus has to wait for the cell to actually transcribe the viral genes, and that might take some time. But and I'm sorry, the viral genes must be transcribed into RNA, which can then be used to create proteins. That's because the material was DNA. The DNA needed to be transcribed to RNA first. But RNA viruses skip that step. They just start making a bunch of RNA sequences, and those RNA sequences are immediately translated into new virus materials and parts. Okay? RNA is worse. RNA is worse. Well, RNA just wants to work faster. I know I said that lytic infections are fast, but they're fast compared to lysogenic infections. These RNA viruses, they happen even faster because they're cutting out that extra step that the lytic and lysogenic infections take, which is they need to inject DNA. The DNA is then transcribed by the cell into RNA, and then the RNA can be used to make the new viruses. The RNA viruses just release the RNA and say, okay, start right now, and the cell does. So RNA is faster than DNA, yeah. That's why it's really quick. It's really easy to catch a cold from someone. You'll know who you got sick from if you were around a person that had the common cold, because it acts pretty fast. And the reason why here, actually, I'll highlight this right here. I mentioned the ribosomes earlier. Those are the organelles inside a cell that construct proteins. And the host cell's ribosomes sometimes mistake the viral RNA for the host's own messenger RNA or mRNA. And it's just the cell is just completely clueless about what's going on. It has no sort of response to this happening. Okay, so that's one type of RNA virus. Let's talk about the second group of RNA viruses, which we call retroviruses. Now, excuse me. So what happens in a retrovirus? A good example of a retrovirus is the human immunodeficiency virus, or HIV, which leads to acquired immune deficiency syndrome, or AIDS. And I'm sorry, I said autoimmune deficiency syndrome earlier, so I got that one wrong. But yeah, it's acquired immune deficiency syndrome. So this virus works a little differently, okay? So it doesn't release a bunch of copies of RNA like our cold virus does. What it does is it releases one copy of RNA right here, and then what happens is enzymes from the cell come over and they see this little bit of RNA floating around. They're like, this thing must be lost. And the enzymes come over and they make a DNA copy of that viral RNA. So here, right here, you can see this little yellow enzyme or protein or whatever it is. Well, if it's an enzyme, it is a protein. This little enzyme here is it's constructing a DNA version of the viral RNA sequence. So it's like, oh, I'm sorry, buddy, you're lost. Let me help you out. Let me fix you up some DNA and then take you right back to the host's genome. Wow, talk about disaster. So that viral DNA that has just been made is then taken to the nucleus and it just inserts nicely into the host genome, similar to our lysogenic infection. Remember, the viral DNA from the virus the viral DNA in a lysogenic infection actually kind of fits nicely into the host's own DNA. Well, that happens here. It's just, except this time, that RNA virus, it needed to get the RNA transmitted in, or translated into DNA before it could be inserted into the host genome. So, a lot of different sort of orders of how things are taking place, but the gist of it is all the same. Okay. So, what happens when the viral DNA is now in the host's genome? Well, again, it might take a while. This might happen over a long period of time. That's why it takes a long time for acquired immune deficiency syndrome to develop once you have contracted the HIV virus, because it takes a long time. The virus has to wait until this particular sequence of its DNA is opened up, unzipped, and then transcribed, and then used to make viral parts. So that might take a while, depending on how frequently the cell is accessing its genome. How often transcription is taking place. So this is a much longer term sort of infection. But the same thing happens. Eventually, it is transcribed, and that transcription is then translated into virus parts. And then the viruses are assembled, and then there's a bunch of them, and they rupture the cell from the inside, and they escape. So all viruses, their cycles end the same way. They rupture the cell, and they escape, and they try to infect other cells. Does this mean the chest burster is a virus or a parasite? I would argue, actually, Adam, that it is parasitic. It is parasitic. However, it is not a true parasite, because if you guys have never seen Alien, um, let me fill you in on some information. uh, so Alien, or xenomorph face hugger. So this is how a xenomorph reproduces. Uh, they have these sort of transitionary life forms, which is a face hugger. And the face hugger carries fertilized, I'm guessing it's fertilized eggs. They hatch from their own eggs, but they carry fertilized eggs from the alien queen. And then they they latch on to a suitable host's face, and they inject their fertilized eggs into the host's stomach, where they embed, and then what happens is that fertilized egg will hatch into probably some kind of larva, and the larva then feeds off of whatever the host is eating and grows and grows and grows. So in that sense, it's parasitic, but eventually it has to leave the human body once it's fully developed, and it needs to start feeding on other things. And in that sense, it is not truly parasitic. In the case of a chest burster, and once it bursts out of a person's chest, that person is then a what we call it, a transition host, meaning that's not the end that's not the end destination for the chest burster. Its end destination is to move around and hunt, and the parasitic part of its life cycle is only temporary, um, because parasites don't usually kill their hosts. That's the main thing there. I just went into I just went into a huge discussion about freaking chest bursters in my science class. That's crazy. Okay. We already went over this chart. We kind of went over it. I mean, basically we described the characteristics that viruses share with living things, and I'll highlight them here for you guys right now, or I'll bold them. Oops, did not mean to do that. So a virus is not made of cells, okay? we can just rule this out right here. Um, they do not move or use energy. They move through vehicles and vectors like animals or they move through water or wind movements. They don't move on their own. They do have genetic material. They do reproduce. They do respond to stimuli. I mean, they evolve over time. We encounter new strains of viruses every single year. That's why people recommend getting vaccinated for the flu every single year, because after a year's time, the virus has probably changed in some way. But they do not grow or develop. Okay, so these are the three sort of characteristics that viruses have in common with normal cells. But these main ones here, they do not exhibit, which is why we cannot classify them as living. They're these non-living things that constantly try to seek out and kill cells. It's a very scary premise, almost like a movie, but it's real life. So have fun sleeping tonight, guys. Is what I'm saying. All right. What was I gonna do? There we go. So I wanted to show you guys a short video about the coronavirus and how it infects human cells. So we can take a look at it, and then based on what we discussed today in terms of our RNA viruses, the different methods of infection, and our DNA viruses, our lytic and lysogenic infections. Let's see if we can compare what we learned today about these infections to the way that the coronavirus infects human cells.
Researchers from China have used cryo-electron microscopy to show how SARS-CoV-2 infects humans.
Wow, talk about mono audio. My I can only hear it in my left headphone. Huh. My right ear is lonely.
Researchers from China have used cryo-electron microscopy to show how SARS-CoV-2 infects humans. The study published in Science says the virus targets a type of receptor found on human cells in the lungs, heart, kidneys, and intestines. Citing Kingston University microbiologist Mark Fielder, Sky News reports that the virus seems to attack two types of lung cells.
Okay, I'm going to pause right there and I'm going to go back to the textbook really quickly. So, remember those surface proteins I was describing as being unique to every single virus? And even the shape itself is very different. The reason why uh, all these different viruses have different surface proteins is because those surface proteins act as sort of like a fake key, so to speak. Um, they'll try to make contact with the cell wall, the cell membrane, and the surface proteins will sort of signal to the cell, "Hey, let me in." So that is why viruses only target very specific cells, because they only have the surface proteins, the fake keys, to trick very specific cells. I mean, so if the influenza virus tried to attack, let's say, some one of your organs in the digest, maybe it tried to attack your stomach, it wouldn't be able to infect your stomach cells because its surface proteins only fool cells in your respiratory system, if that makes sense. So every virus is sort of looking for a specific type or a couple of types of cells to infect. Because those are the ones that can fool, but it can't fool cells that it doesn't have these surface proteins to trick. So let's go back to the video.
Goblet cells that coat the respiratory tract with mucus, and ciliated cells that usually filter out pathogens. A previous study published in Science found the virus spike protein has two receptor binding domains, or RBDs, facing downward.
So those RBDs are basically where the virus actually kind of that's essentially the key, right? So it binds to the receptors, those specific receptors, and then it tricks the cell into letting it in.
And another facing upward. These allow the virus to bind with and invade human cells. The virus targets a human ACE2 receptor that has bonded with an amino acid transporter. This subtype of ACE2 structure has never been discovered before. The virus uses the spike protein's up RBD to penetrate the cell. The virus then dissolves its own protein shell and releases its RNA payload inside the cell, according to British Society for Immunology. A study in Frontiers in Microbiology says a coronavirus hijacks the cell's structure to reproduce. The viral RNA takes over the host cell's endoplasmic reticulum to replicate itself and to manufacture the protein parts to make new.
So I'm guessing it attacks the rough endoplasmic reticulum because the rough endoplasmic reticulum, if you guys remember, is dotted with ribosomes. So that's interesting. It goes straight to the endoplasmic reticulum. It doesn't go into the nucleus apparently.
New viruses. According to the Society for Immunology, the hijacked cell's Golgi bodies then package viral RNA and proteins in a viral protein shell. This leads to the creation of new viruses that leave the infected cell via the membrane. The study in Frontiers in Microbiology says coronavirus takeover imposes stress on the host cell. Cell death or apoptosis is the result when the infection overwhelms the host cell's ability to maintain homeostasis.
So that's interesting. Okay. So basically, this virus operates a little differently. It's an RNA virus, and it makes sense. Most coronaviruses, I think all coronaviruses are actually RNA viruses. Let me go back to the chat. There we go. So the virus it kind of fuses with the cell membrane. That's similar to endocytosis, if you guys remember. Let me look up endocytosis. That's a form of active transport. If you remember, let's see, where is it? Where's the reverse one? Well, basically, I guess I'll look at the reverse copy. So if this is the virus and this is the outside of the cell, it comes in and it fuses with the membrane and releases its RNA on the inside. So interesting. Okay, and then the viral RNA from the coronavirus then is immediately translated in the endoplasmic reticulum, and then all the proteins and nucleic acids that were built are sent to the Golgi apparatus, which is the part of the cell that kind of packages things together, and that's where the viruses are assembled. So actually pretty interesting. So it is an RNA virus and it is not a RNA to DNA virus, so it does not act in the same way that the HIV virus does, where its RNA copy, its viral RNA is made into DNA. It's just immediately taken into the cell's endoplasmic reticulum and translated. Wow, pretty interesting. Okay, that'll about do it. I think I don't really have anything else for you guys. Let me pull up the Google Classroom right now. Let me get rid of this stuff. Chest bursters. Um, Here we go. Okay. Attendance check. Let's see. Who is here right now in the chat? I know Nat is here. I saw her post a very nonchalant comment. What'd she say? The fact that there's no cure is what's dangerous. Yeah, I guess the binding mechanism of this particular virus is tricky to sort of I guess synthesize a defense against. Um, yeah, there's other viruses that spread quicker. I mean, influenza spreads very similarly. The reason why it's really tricky to stop the spread of coronavirus is because this virus is made incredibly fast. I mean, and we saw it makes sense that it's not similar to our HIV virus. It doesn't act that slow. So it's definitely not being inserted into our own DNA. Instead, it's just being immediately taken and translated into new viruses. So it happens really quickly. And yeah, I hope that I definitely hope that they can find a way to kind of counteract some of those viral mechanisms soon, so that we can get out of the situation. So let's get back to this. Okay, Sammy's here, Connor, Adam, Bryce, Michael, Natalia's here. Anyone else who I didn't really see their name pop up? Nathaniel, is Nathaniel here? If you guys came a little bit later while we were already in the middle of class, go ahead and post your favorite holiday movie on the Google Classroom page. Is Christian here? I really hope he is. I'm gonna stop recording right now. And while you guys are doing attendance, I'll just go back. Remember, you guys are doing 20.1 tonight. I want it. I want it done. I want it turned in tomorrow. This is the last thing I want you guys to do before the break. Uh, Bryce, I don't think it's that coronavirus is more dangerous. Not that the symptoms itself are more dangerous. What's dangerous about it is how easily it's transmitted. Um, the reason why we have these social distancing and 6 feet kind of apart rules. I mean, if you guys have been to a grocery store recently, they have like little mats laid out to tell people where to stand when they're waiting in line. And the reason why is because it can be transmitted simply by breathing. Enough viruses are created uh, that the cell, well, enough viruses are created that it's all it takes is breathing for those viruses to be ejected into the moisture in your breath, and then transmitted to another person's respiratory system. Uh, so that it's just more it's more infectious really. So more people will get it. And yeah, flu is also transmitted through coughing and sneezing and breathing, but uh, not enough viruses are transmitted for every breath and every cough and every sneeze for it to be as infectious as COVID-19. I mean, we saw in the video actually how the cell doesn't immediately rupture. The cell doesn't immediately rupture. Actually, what happens here in this part, if you guys remember, let me mute this. So here's the viral proteins and nucleic acids being constructed into new viruses by the Golgi apparatus. And here's what happens. These viruses don't build up inside the cell. They just start leaving immediately. Okay? And only once it takes a while until the viruses build up that they rupture the cell. So there's already viruses that are leaving the cell without killing it. So that could explain why there are so many viruses found in the infected individuals, is because the cell can produce more viruses for a little while longer before it eventually dies, and the viruses are still leaving while this is all happening. Okay. Uh, Natal, yeah. So it just keeps producing it, like we saw right here. The viruses are being constructed and then leaving as soon as they're made. So they're not building up from the start. They're already leaving. But I'm guessing there's only so much. Remember, we're limited by the surface area of the membrane. So a virus starts streaming out until eventually there's not enough open channels for them to leave, so they start building up, kind of like a long line forming, and then eventually it bursts. But and before it bursts, there are still viruses that are leaving through the cell's different channels and pumps. Well, I'm guessing mostly channels. There is no extra credit during vacation, Bryce. We're just going to do this assignment, 20.1, and then we're going to come back and focus on other things. So we're not going to go into the rest of Unit 20. We would the lesson today on viruses. Just I thought it would be cool. Um, any other questions, Natalia? Ask all your questions right now. General Grievous got coronavirus because he coughs all the time. It's really interesting that he coughs all the time because he's a robot, right? Okay, yeah, we'll be going into science fair when we get back. So this is sort of the end of our Unit 20 foray. How long are we going to be stuck in house? Didn't Garrett say that they announced school closure until the end of the year? Let's see. LUSD. Coronavirus shutdown worsens student inequity. Many of them. Oh, jeez. Yeah, it looks like it seems they haven't made the announcement to end the school year, but it looks like they are definitely there are people there who definitely want to close school for the rest of the year. Your mom? Oh, really? They're closing until the end of the year? Oh, jeez. Well, that freaking sucks. Well, hold on a second. So LAUSD decided. Okay. All right. I have to read up on that. This is unfortunate. Okay. I am. I'm gonna go cope with this new set of news right now, but I will still be checking my email, so please reach out to me if you have any questions or trouble with the assignment. So I'm gonna end the stream right now. Thank you guys so much for coming. Hopefully you learned a little bit about the coronavirus today and just viruses in general. And moving forward, have a great spring break and come back ready to finish science fair. Uh, Bye everyone. Have a nice day, and yeah, I'll see you when you get back. Take care.
Sometimes I feel like I don't have a partner. Sometimes I feel like my only friend is the city I live in, the city of angel. Lonely as I am, together we cry. I drive on the street